we don't have a class on next Thursday, so I'm not sure about the regulation for next Thursday. And right, completely correct. No, there's no class next Thursday. There is class next Wednesday. We moved it from next Monday to Wednesday. Anyone? Yeah, next week instead of Monday. I mean, we are all here on the mailing list, right? So for sure you had to at some point read about that. So instead of Monday, we will do the next week class on Wednesday from 2 to 4 p.m. And sorry for this change. Anyone who can't make it, I will, of course, if anyone will want to sit together after, we'll find a time to sit together and go over the material together.
So, sorry, but I didn't see the mail yet.
Yeah, sure.
So, on what hours is it?
2 to 4 p.m. afternoon.
So, we don't have classes until the night? No. Okay.
This is when most people reply that they can do it there. I send it as a doodle it's called, doodle poll, something, yeah. Anyway, it's next Wednesday, 2 to 4. Anyone who can't come, it's my responsibility to fill him in, okay? So, yeah. Do you want to do, by the way, today, skip the break and finish at 12:30?
Yes.
Okay, let's start and see how we go. Let's start with your question page about transport ions and cross-cell. No, sorry, not this one. My mistake. The architecture, of course. This is the chapter we finished last time. I opened the wrong one. Second, cell architecture. We still didn't answer this question together, right? Because we only finished it last time. Okay, so for the first question, some organelles in the cell are surrounded by two membranes, and in between these membranes there is an Did we answer these questions together already?
I don't think so.
I don't think either. But all of a sudden I have a deja vu from last year. Okay, sorry. Okay, let's start again. Some organelles in the cell are surrounded by two membranes. It's a bit weird, but because most of them are surrounded by just a single membrane, but some of them are surrounded by two. And in between these two membranes there is an intermediate space. Who are these organelles that are surrounded by two membranes instead of just one? Nucleus and mitochondria, correct. So this is B. So nucleus is the two membranes there fused together to form the gate, the pore, the nuclear pore, and for this, no. And mitochondria also has intermediate space between the two membranes. We saw that in electron microscopy figures. Right. Phospholipids are distributed in a heterogeneous manner between the two layers of the plasma membrane. Now we mean just two layers of a single plasma membrane. How does this heterogeneity achieve? Either by different organelles in the cell synthesize phospholipids which arbor in one of the two layers. It might be true, but alright. Different enzymes between the two sides of the membranes cut different bonds in phospholipids hydrophilic regions, and then change their structure and their identity, and so there will be different phospholipids in both sides of the layer. Which do you think is true? We actually mentioned both as possible explanation.
What's the difference between A and B?
So A is the organelles that synthesize the phospholipids themselves, and one of them, the ER, will synthesize phospholipids that will eventually find their way to the exoplasmatic side, and the Golgi will do the same thing for those that will desoxyrize to the the other way around? No, I don't think it's the other. For the inside of the cell. So these are two different pathways of phospholipid synthesis, and one of them produces exoplasmatic phospholipids, and the other one does not. We have mentioned it. I think it's also in the slide. And the second explanation is that different enzymes between the two sides of the membranes cut different bonds, and then change the population of phospholipids that does ring a bell? B.
When you say that they cut different bonds, means there are phospholipids already there, and then the enzymes change them? Is that what it's saying?
Exactly.
Okay.
Enzymes change them and change the ratio between the phospholipids
Mm.
across the population of phospholipids. So that the explanation of why do we have heterogeneity in the phospholipid population actually turns to a question of why do we have heterogeneity in these enzymes population, but we do have them across the two sides of the biomembrane. Right? So sorry. So A and B is true. Both are true. Both are true. Different organelles in the cell synthesize phospholipids, which will be either in the outside or the inside side, and different enzymes. So just go back to the slide. I think it will be clear because I think both explanations are there. A and B are correct. The answer here is B. Okay. Question number 3. Below is a list of cellular organelles. Match one organelle or more from the list with its role in the cell. So these are the organelles. These are the these are the roles. Let's start with degradation of outside material and all cellular organelles. Sorry? Which of these organelles is responsible for degradation of outside material? Lysosomes, right. This is where this degradation takes place. So this is lysosome, ATP synthesis by glucose oxidation. Mitochondria, correct. The power plant or the energy plant of the cell. Synthesis, processing, and sorting of secreted proteins. RAF-VR with an emphasis on the synthesis. If the synthesis is there, so it's for sure the RAF-VR where the ribosomes are located. Storage of secreted proteins, and now we right, secretory vesicles. We only talk about storage this time, so this is secretory vesicles, correct? Regulation of entry and exit of molecules to and from the nucleus, obviously the nuclear membrane, yeah, membrane. Okay. So we finished, we stopped last time while discussing voltage-gated ion channels and the propagation of action potential in nerve cells, and we went over this slide that shows the really seminal experiments that prove the inactivation of voltage-gated potassium channel. Let's go on with the nuance of how the action potential is propagated actively through not exposed, unmyelinated neuronal projections, but through or along myelinated axonal projections. But before that, just one quick answer. We discussed last time also, we looked together at these traces, which are patch clamp traces, and I consulted Mickey, and indeed, even in patch clamp, when you zoom in very deeply into one of these time frames, you will be able to see the refractory period, obviously. So these lines here are linked just because we are zooming out so much. In the level of a few milliseconds, we will see the refractory period. Of course, this is a phenomenon that occurs at the single channel level. And this is exactly what patch clamp is. So for sure, we will see the refractory period here. I've also been asked 2 classes ago about RNA termination, synthesis termination, how mechanistically it occurs. I think we are too much out of this context already. So I have the answer, but I will go back to it when we'll discuss post-transcriptional regulation. So it will be more linked to what happens to RNA after it is synthesized. And then I will go back to mechanistically how does the synthesis of RNA stops. Again, mechanistically, I'm not just saying that there is a stop codon, but exactly what happens between several proteins and the RNA molecule that happens that induces termination of synthesis.
So if patch clamp is a single channel experiment, how come there are two?
So these are the cases in which you caught accidentally on a piece of membrane more than one. It's true. So the more accurate way to describe it is a single or just a few channels per experiment. Okay. So we have described the active propagation of nerve cell of the action potentials along nerve cells, but for these myelin covered axonal projections, the case is a little bit different. There is no active propagation throughout the axon, but only in these exposed sites. So these are the covered myelinated regions, and these are the exposed sites that are called nodes of Ranvier.
There's a band called that.
Really?
Yes.
I didn't know. Okay, so exactly in these nodes of Ranvier sites, this is where you will find high concentration of the voltage-gated sodium channels, the ones that are responsible for the active boosts in the amplitude of the action potential. Okay, so this is what happens only in the nodes on Ranvier. So inside or along these covered regions, we do not have any active propagation, as I said, but we do not have any decrease or substantial decrease in the amplitude of the action potential as well. So don't worry. So it's sort of, if you like to plot the action potential from here to there, its height will diminish in about 1 to 2%. This is really nothing, and then we gain again these the amplitude right here. Okay, so how do these channels, voltage-gated sodium channels, are concentrated? Why do they concentrate in these regions? For several reasons. It occurs via interaction with a family of proteins that are adhesive proteins. It's called NR-CAM. So they can look at them as sticky proteins that adhere to other proteins and cause their aggregation. Glial cell, okay, so first of all, the myelin sheath, I think we have mentioned it already. The myelin sheath is actually the plasma membrane of another cell, not the neuron itself, but a glial cell, a supportive cell, which adjacent cell, and it comes and right to the axon, and it covers the axon many times with its plasma membrane. So these cells, or adjacent glial cell to the nodes of Ranvier, will also secrete protein hormones that will trigger the clustering at these nodes. Several intracellular processes are triggered by these hormones right here inside. We will not mention them in detail, but just saying that they exist. Also tight junctions, which are something like molecular stitches between the membrane of the glial cell right here and the membrane of the neuron right here, form this isolated regional island, because you remember that the plasma membrane is fluid, and if you put something here, it will flow around and might leave this side. But if you close it with these tight junctions all around, so the sodium channels will stay exactly where they were located to begin with. Okay, so these stitches can be found immediately adjacent to the nodes. Right here, this node, by the way, is called the paranodal junction. Paranodal, just right directly adjacent to the nodes of Ranvier. This is the paranodal area. Okay. And along these nerves, what we have is what we call saltatory activation or saltatory excitation of these nerve cells, because the action potential gets its boost right here, and then the next one happens right here, and this sort of these jumps reminded of saltatory activation. Okay. Now for our last point in this chapter, we will discuss the principal components that we find at the synapse site: neurotransmitters, receptors, other transport proteins. All of them can be found in synapses, presynaptic and postsynaptic cells. Okay, so first, let's say that neurotransmitter receptors can fall into two broad classes. The first one is the one that we will discuss in this chapter, the ligand-gated ion channels, which are channeled by themselves. They open immediately upon neurotransmitter binding. The second kind is usually G-protein coupled receptor. These are receptors that do not have any channel that will open or will stay closed in them, but they induce a different sort of change, and they will induce indirectly the opening or the closing of a separate ion channel. So this will be their effect, but it will be an indirect effect. And this will be the topic of our next chapter. And hopefully, we will be starting also today, the G-protein coupled receptors. But for now, let's focus on the ligand-gated ion channel. Okay. Neurotransmitter they are simply amino acids. Most of them are simply amino acids, the same substances, same molecules we discussed in the context of proteins. Here's an example. Glycine is an amino acid that can be found in many proteins, but also a neurotransmitter in its unbound state. Glutamate, same thing, very important neurotransmitter. Derivatives of amino acids. Famous examples are dopamine, norepinephrine, and epinephrine. All of these three are derivatives of the amino acid tyrosine. It would be a good time to go back to the slide where you see tyrosine unchanged and then see exactly the change that occurs when it becomes a dopamine or norepinephrine. We can appreciate the change between neuropinephrine and epinephrine right here. An additional carbon here, one carbon less is what makes neuropinephrine different from epinephrine. Also called adrenaline, the stress hormone. Okay, serotonin derived from tryptophan, histamine derived from histidine, and GABA, again very important neurotransmitter, is derived from glutamate, which again can be a neurotransmitter by itself. Other neurotransmitters such as ATP also exist and outside of this slide. Okay, so you will hear a lot in physiology courses about GABA and glutamate and so on.
ATP is adenosine?
Yes, adenosine 3-phosphate. So when they lack phosphate group, meaning in their state of adenosine, ADP and AMP monophosphate, adenosine monophosphate, for instance, can be cyclic AMP can be a neurotransmitter. In next week, I'll mention it in later on, but in next week we will read a Kandel article, very important brain scientist, where cyclic AMP is a neurotransmitter. So, okay, so not only not only amino acids, but also small molecules such as AMP, cyclic AMP, should say. Okay, there is some, I should mention here, there is some disagreement or overlapping definitions between what is a neurotransmitter and what is a neuromodulator. Maybe you already heard about neuromodulators. I think the agreement between scientists is that neuromodulators are secreted in a more distributed manner, and not per synapse, but usually per several adjacent synapses. Okay, so this is one change. And the second change, more maybe modelistic approach, would be that it looks like they change the gain of the synapse and not their function per se. Which means that if you have this neuromodulator around, so upon activation by a neurotransmitter, you will have higher or lower activation or response to the neurotransmitter. So this would be the effect of a neuromodulator, changing the synapse's gain. Okay, but we'll not go into this right here. Each neuron generally produces just one type of neurotransmitter. And this is why, again, you will hear a lot about cholinergic neurons, GABAergic neurons, and so on and so forth. And that means that we are talking about neurons that produce this neurotransmitter, not necessarily about neurons that respond to cholinergic signals, for instance, or GABAergic signals. Okay, again, a neuron can respond to a cholinergic signal and produce GABA, and then secrete GABA presynaptically, or from its presynaptic side, to affect the downstream or the next in-line neuron. Right? So, per definition, neurons are defined by the neurotransmitters that they themselves produce. Period. Reservation. Some people see neurons that produce more than one neurotransmitter, but this is textbook, okay? And more than one neurotransmitter is in the the literature still. So this is how they told me to present it, okay? There are some articles that showing the neurons can produce more than one neurotransmitter. Right, I said it, okay. Neurotransmitters are synthesized in the cytosol and then imported into membrane-bound synaptic vesicles within the axons, okay? So again, we're talking about the presynaptic site, and there they are stored. They are roughly 40 to 50 nanometer in diameter, and the lumen has a low pH, meaning lots of hydrogen ions. Okay, and this low pH is produced by V-class proton pumps, which were discussed last time. These are the classes the pumps that use ATP hydrolysis to pump hydrogen inside these vesicles. Then the next step is to use this proton concentration gradient to import to import neurotransmitter into these vesicles, and this is a process that is done by ligand-specific proton-linked antiporters.
Why?
Weird. So why it's a good question? Most biologists would just say that this is, evolution for evolutionary reasons, meaning that so again, the question is, if I understand you correctly, so why not instead use ATP hydrolysis to pack neurotransmitters right into the vesicles, and instead of doing this unnecessary step by using ATP to pack hydrogen, and then taking an hydrogen out to take a neurotransmitter in? I'm not sure if there's a good answer, but again, when biologists see these processes that are not the most efficient that one can think of, so they will argue that this process preceded in evolution this process, and then when the need for packing of neurotransmitter into vesicles have emerged during the evolution, so it already used what already existed, the preceding process. We see that a lot in biology, and this is the consensus explanation. Yeah. But I agree that it's not effective energetically, but still, I think we'll see another example of not most effective biology even in this chapter later on. Okay. We'll see here a specific example from the cholinergic system for co-regulation of components in the neurotransmitter system. That is actually, so this is a specific example. I'll say the details in a minute, but it's really an example of a more principled global concept in neurotransmitter regulation or neurotransmitter system regulation. So, acetylcholine is the neurotransmitter, and it's produced from these two reactants, acetyl-CoA, and choline. And the enzyme that promotes this chemical reaction that produces acetylcholine is choline acetyltransferase. Okay, so this is one side of the story. And then we have the antiporter, proton acetylcholine linked antiporter. As was mentioned right here, it sits on the vesicle membranes and take one hydrogen out. This is what gives it energy, the process energy, to take one neurotransmitter in against its concentration gradient. Okay, as any other antiporter will function, and it's called the hydrogen acetylcholine antiporter. Now, the gene that encodes for the antiporter is contained entirely within the first. So we haven't defined introns, but never mind the introns. The gene that encodes for this protein is contained entirely within the gene that encodes this protein, the choline acetyltransferase, in the genome. So just simply in the sequence of nucleotide sequence in the genome that codes for this protein is contained. It's really inside the sequence that codes for this protein, and the outcome is that these two usually are transcribed and then translated together. And it makes a lot of sense, because if you want to have more packed cholinergic vesicles, so if you will just increase the level of the choline acetyltransferase, you will have a lot of acetylcholine, but unpacked, and so unready to be released and induce cholinergic transmission. And the same and the other way is to look, if you have more than antiporter, so this will be a bottleneck. If you want to produce more acetylcholine molecules, you will not have anything to pack, right? So only when the two are synchronized, it's an effective process.
So when you say they're synchronized, you mean they have the same promoter?
Yeah, so they sit in the genome of the same promoter. I wasn't sure about mentioning the promoter because I haven't defined it in this class, but yes. They sit on the same promoter in the genome, exactly. Yeah. Okay, so this is one example, classical, very, very well established. Other processes to co-regulate these different components of the same neurotransmitter systems also exist, sometimes at the level of RNA already. Right, so this is already at the level of the genome, but other co-regulation of neurotransmitter systems happens post-transcriptional at the RNA level.
I got the work that
So the hydrogen is packed by a V-class pump into the vesicle, and it's high concentration inside, low outside, used to take it out. So this is an energetically variable process. You will gain energy by taking a hydrogen right here out, and this energy is used to take a neurotransmitter against its concentration gradient.
Okay, this is connecting and the other part is just transferring it into the cell.
What do you mean by connect? Yeah, it links these two molecules, produces acetylcholine, and then acetylcholine is the red molecule right here, the neurotransmitter. Yeah, that is packed into the vesicle. All right. Okay. And we will give more specific examples from the cholinergic systems because this is what we like best and know best in Monad Lab. But these examples are really
So
a lot of examples.
that mechanism with the anti-porter pump, so is that a reuptake mechanism?
No, not at all. So reuptake is from the outside of the cell. I mean, reuptake is just a word, but when in synapse we use it to say taking a. So we will. You know what? The next slide we will see reuptake. So, all right. Okay. So the neurotransmitters have been packed into these vesicles and are now we are still in the presynaptic site and they are shuttled towards the presynaptic site. The axonal projection can be a long way, but still they are shuttled to be spatially proximate to the presynaptic end. Okay, so these are these vesicles, and now experimentally they are defined, or they were these definitions of first and second populations of packed vesicles are simply because of experimental reasons. You can trace the articles that started naming them this way, but we stick to this definition. So the first population of packed vesicles are just not anchored yet on the plasma membrane. Just wait to their turn for their place to be anchored. And the second population are those that are already anchored by these unique specific proteins here. So these are in a standby state, and these will take longer for them to be able to secrete their content, meaning the neurotransmitters. Now it's a common mistake to think that the action potential per se or by itself is the abrupt depolarization that flows through the axon and reaches the synaptic presite. Is what drives or push the packed vesicles outside of the cell and then cause neurotransmitter release, but this is not really the case. The case is that the depolarization, meaning the action potential, affects first these proteins. Sorry, not first, of course. Affects first these channels, which are voltage-gated calcium channels. Then calcium flows into the presynaptic site from around the synapse into the synaptic site. Calcium induces many things. Among them, it changes the conformational state of these proteins, the ones that anchor the synaptic vesicles to the plasma membrane, and that causes them to fuse and the neurotransmitters to be secreted to the synaptic left. Okay, so the calcium here is an important link. Okay, so that happens, and then what we have. Okay, then we will in a minute we will continue the journey of the neurotransmitter across the synaptic left to the prosynaptic site, where it will meet and be recognized by its receptor. But first, what we have in almost 100% of the synapses for any given neurotransmitter kind, what we have is a reuptake process of the neurotransmitter back to the presynaptic site. Not necessarily the same way it was secreted, meaning that, for instance, for acetylcholine, the acetylcholine itself is break down to two different molecules at the synaptic left, and only part of it, one portion of it, is taken back with the reuptake process. And for glutamate, for instance, there is the reuptake of the whole molecule, the whole glutamate. But for all of them, what we have, the reuptake process is performed by sodium-linked syn-porter. Why does it make sense to use? Why is it a syn-porter? Because sodium is high outside, right? So taking sodium inside is the energy that's coupled to taking the neurotransmitter back inside, against its concentration gradient for the neurotransmitter, and down its concentration gradient for the sodium. One more minute. So this is the reuptake that happens in a neurotransmitter, something very similar, which we just call a little bit differently. We call it recovery.
of the synaptic vesicles happens for the vesicles themselves locally, and this is done by a protein called clathrin. It suck or engulf the vesicles back into the presynaptic space. Yes.
They have to go back to vesicle? Like if after they break with the acetylcholine
Yeah.
breaks and only the choline comes back in?
Right.
And
So then there will first be synthesis of the choline back with the
before
acetylcholine transferase before it will be packed. But okay, yeah,
Okay, no,
yeah.
okay. And can you explain again the sodium, why sodium is
So you might think almost for all neurotransmitters, the reuptake is a sodium dependent process. It happens with these different symporters exist, but all of them are symporters using sodium concentration. It only makes sense because sodium is high outside and low inside. Well, you could have done it with the potassium as well, I guess, taking out the potassium, but
Oh,
yeah.
okay.
But
Choline a negative ion?
sorry?
Choline a negative ion?
No. So what if you're asking, is it a negative ion by no? No, it's not. So what you're asking is whether this is an electron-neutral process
Yeah.
or not. So usually it's a good question. Usually this process is linked with either entry of a negative charged ion. So these are usually done by non-gated chloride channels, or taking out a positive ion. I think is less frequent. But taking for antiporters and symporters, the same case we saw here when we tried to build up the concentration of these positively charged hydrogen ions. So we had to open a tap for a negative charge to flow in. So same thing happens here. Negative charge does flow in through non-gated chloride channels. And then the net process is neutrally electroneutral.
So,
Just you don't see it here, but
so in the symporter you get to keep the concentration by letting out the sodium?
the charge
ion?
concentration. You don't
The what?
the charge concentration. This one sort of destroys the sodium concentration, but you do have ATP powered pumps that push sodium, take out sodium all the time. Again, the functions are in the background; they're not here. But
So can you say again why do you need to take out one sodium ion, take out one choline?
we take a sodium back into the cell. It's not like we need or don't need it. Just that if we'll place a symporter here, so this will be the only way it can function, taking sodium in order to, which is down its concentration gradient, gives you energy to take neurotransmitter in. Against its concentration gradient, you have to pay energy.
Okay.
And in parallel, you have to let one chloride in as well. Chloride ion. Does that make sense? Did I answer what? This is presynaptic site, synaptic left, postsynaptic site is down there. Okay, and then the uncovered vesicles, uncovered by clathrin, are packed again with the neurotransmitter. This whole loop, in different synapses it takes different time epochs, but I think on average what I hear is people talk about approximately 60 seconds, approximately 1 minute for a vesicle to secrete its neurotransmitter and be recovered and pack again with neurotransmitter. And I guess it really varies tremendously, but 1 minute is yes, all right. This process looks very much like exocytosis. This part, exocytosis, and this is endocytosis. We have mentioned endocytosis in the past. Exocytosis, I'm not sure, but it's basically the other way around as endocytosis. Substances are taken from the cell towards outside, and so it's a general mechanism that happens in all cell types. But in neurons, so even I admit that there is some unique things in neurons. So in neurons, we have two unique features for this exocytosis, endocytosis. The first is that the exocytosis is, or the secretion is tightly coupled to the arrival of an action potential in the axon terminus. Okay, so that is unique for neurons, and the synaptic vesicles are recovered or recycled, same thing, locally right here. Well, it's not action potential per se, but also entry of increased level of calcium. Several intracellular mechanisms.
So it's the
It
same
usually
process,
involves
but
calcium.
it's just
Yes.
not caused by
Almost the
action
same
potential.
proteins, absolutely. Almost the same proteins, sometimes shared proteins between exocytosis that happens in neurons, these green dots or whatever. So same green dots you'll find in the liver cells. But again, okay, so there is some overlap between, okay, but still some unique proteins that only can be found in presynaptic site of neurons.
So they get some kind of voltage change, but not due to action potential, due to chemical change?
Exactly, not an electrical signal, but well, again, a reservation, pacemakers in heart, cells, smooth cells, all of these, and other tissues, I guess, I just can think of, right here, but also use electrical signals. It's not unique to neurons at all. But an action potential, this huge spike that goes up almost 100 millivolts and then goes down, and all that happens in a time frame of 2 milliseconds, or something like that. So this is unique to neurons. It's sharp. Yes, sorry.
And what happens in other cells about recycling of vesicles?
It's just that it's not locally, and it's not the same. When we say that the same vesicles are being recovered instead of new vesicles being formed, which is the case for other cell types besides neurons, we actually, so after this synapse, this vesicle here was fused, so there is no vesicle anymore, right? So, but there is a vesicle yet, still, because these proteins are not detached from the membrane. So this whole complex of proteins is what we refer to right now as a vesicle. And they will form again the vesicle that, locally. And this, for neurons, the outcome is that it takes much less time than any other cell types. Sure.
With all these things, do we have some models that model their dynamics? They're like Hodgkin models?
Yeah, so it's not, I'm not sure about model these, when you model something, so you model an experimental result. You do not necessarily care all the times if it comes from this process, or it's differently altogether, the slide will look differently and still the readout will be the same, and you actually model the readout. You want to get the same profile.
I understand, but
Yeah.
I think
Yeah.
specifically that there are many factors here, right?
Right.
Many
True.
regulating factors. If you change one, might be that the process might occur differently, but still occur in some way. So to model that, you need a full mathematical model of this.
Sure. So I'm not sure, I don't have a good answer. So Idan-Segev, but still, what Idan-Segev does is taking Hodgkin-Huxley pretty much and then extend it morphologically. Again, from what I know, I never did these models, but from what I read again, so it takes, instead of one dot in space, so now the Hodgkin-Huxley, it's for each piece of membrane, it's true, and down there's the dynamics of how the signal flows between these different pieces of membrane. But this is still only Hodgkin-Huxley. What you have there is still just channels, from what I know. Again, I'm correct here. I'm not sure how many models you'll find. I guess you could find some that try to encompass more than just channels and put also these regulators into the game. There are some not too serious. Models that are not taken too seriously about whole cell models. Yeah, again, it's not taken too seriously still.
Because we don't know enough.
Because you don't know enough, exactly. Or without the bracket, you just don't know enough. No, because we do know enough, but we don't know enough. When we will have this model, by the way, there will be no animal experiments, I guess, anymore. So, but until then, okay. Yeah.
One more question.
Yes.
Maybe we don't need to talk about it now, but the whole process. Are we going to talk more about the reuptake process and like?
Molecularly? What do you?
Because it's supposed to regulate the whole, like, to give some, so the cell knows how much was secreted and to stop the process, to know when to stop the process, and like it's supposed to have some. The reuptake is supposed to give some.
What?
The cell do something, right?
This process should be regulated as any other process, I would say. Also the packing, no, okay.
What I mean is, okay, so the neurotransmitter is secreted, and then some of it, there's a reuptake that's supposed to be like a negative feedback. It's supposed to be like a feedback loop, so that at some point the cell, like quickly, but the cell knows, okay, I don't know, it gives it some code to do something, knowing how much was secreted or something like that.
Yeah.
Maybe not. This is how I understood it. Like I'm trying to understand in this model.
Yes.
Okay, so what happens now that there's been a reuptake? You're supposed to be.
As a, well, at the, I mean, at the directly or speaking, so as long as you have this link where all this protein what knows what to do with, it can, it shuttles these molecules back into the cell.
Till when?
So now the question, okay, so as long as it is here, but if there is some process that will sequester it, that will take it out of the membrane, for instance, these will be regulatory processes that stops its function. Effectively,
Isn't
we
it
just, yeah.
always like this? Isn't it supposed to happen all the like?
Of course, of course. I
Okay,
mean,
we're just not talking about it?
yeah, we're not talking about it. There's a whole lot, another level of regulation of all of these processes. These will also be regulated. The RNA. I can give examples of, again, for cholinergic transmission, but I really shouldn't here. So this slide is true for all neurotransmitter systems. And what you're asking is actually to go into one example, one of these neurotransmitter systems, and so for cholinergic, there are examples of coupling between. There is even a coupling between clathrin, and I'm not sure, I don't remember what. So we won't go into these details, although they are interesting, and yeah. Oops. Okay. Okay, so as long as we have neurotransmitters here in the synaptic cleft, the downstream receptor will recognize it and function and respond. Signaling the synapses is terminated by either degradation or reuptake of the neurotransmitters. Okay, so if you take out the neurotransmitter back to the presynaptic site, postsynaptic sites, you no longer see the neurotransmitter and stop responding, or you can degrade it, as in the acetylcholine example, at the synaptic cleft. Either case will end with termination of synaptic transmission.
But isn't this case acetylcholine is degrading with reuptake?
True,
Because
also, right. So you have degradation and then reuptake of one of the molecule fractions. You are right. In the acetylcholine case, okay, so this is exactly what we just said. The neurotransmitter is hydrolyzed to give choline and acetylcholine ester, by acetylcholine esterase. So again, in cholinergic synapses, you will find here in the synaptic cleft an enzyme called acetylcholine esterase, and it breaks down acetylcholine and causes it not to be recognized anymore by its downstream receptor, again causing signaling termination. Right, and as you said, the choline is transported back to the presynaptic site. GABA, norepinephrine, dopamine, and all of these very important neurotransmitters all have sodium link importers, as we mentioned in the previous slide, and they are thought to be originated from the same homolog protein because they have very high percentage of similarity, structural similarity between them.
So does the acetyl, does it diffuses to the extracellular fluid or something? Where does the acetyl go?
Yes. Well, it is diffuses from the synaptic cleft and produces a new inside the cell.
Diffuses what?
immune attack.
Sorry?
Immune attack.
The new, yeah, I mean, again, so the acetyl-CoA is diffused from the synaptic cleft. I guess it is degraded pretty rapidly outside the cell, and it's synthesized, and you're not sure where it comes. Acetyl-CoA comes from phospholipid, I think, that is consumed from food. I don't remember which one right now. Yeah, but it's synthesized again inside the cell. Okay, it is not taken in via the reuptake process, as choline does. Yes, all right. Right, and okay, so that we've also mentioned the need to have neutral process altogether, or a net charge of zero, so chloride is often transported with the process of reuptake to remain electroneutral. Okay. Interestingly, and this will just, I guess it will pop up a lot of questions here, but just to mention the fact that, and not necessarily that well explained fact that many drugs, some of them, more artificial, but lots of them really can be consumed pretty directly from natural substances, affect reuptake of different neurotransmitters. These are just examples. Cocaine inhibits the reuptake of dopamine to the presynaptic sites, which cause more dopamine in the synaptic cleft and increase dopaminergic transmission, right? If you inhibit it, and fluoxetine, more well known as Prozac, inhibits serotonin reuptake, and desipramine, again another drug, inhibits norepinephrine reuptake. So again, the outcome, if you add these substances to the system, you will have more of this neurotransmitter in the synaptic cleft and increase synaptic transmission via these neurotransmitters. I'm not sure if there is any good explanation why lots of natural substances or less natural affect these processes. Alright, let's just mention, although we are more interested in neuron-to-neuron synapses, let's just mention the neuron-to-muscle synapse, which is called the neuromuscular junction. Okay, pretty similar to neuron-neuron synapse. We have this synapse, and the neurotransmitter that function in this synapse is always acetylcholine. Right, so this is the function. This is the neurotransmitter that function in synapses between motor neurons and muscle cells. It's a very nice electron microscopy figure where you see the synaptic vesicles packed with acetylcholine, the cleft between the two cells, neuron, muscle, and this is a structure that you learn in physiology, the contractile complex of proteins in the muscle. Anyway, you're looking at a muscle cell. This is, proves that it's a muscle cell and not another neuron. Okay, so this was just an anecdote. Cholinergic, the neuromuscular junction is cholinergic synapse.
Connected to the ganglion?
Not necessarily. Some motor neurons, you mean the spinal ganglion. Some motor neurons come from right from the cortex and innervate the muscle, the peripheral muscle. But this is physiology. We don't care about Christmas or physiology. Well, we do care. Some motor neurons, do not jump through this crossroad junction that happens in the ganglion, spinal, and some of them have transmitter signal to another neuron and just then innervate the muscle. The nicotinic acetylcholine receptor. Okay, now let's focus on the postsynaptic receptors, and again give example of a cholinergic protein, the nicotinic receptor. Again, it's a receptor as for the NMDA example that is named after not an endogenous. Well, it is endogenous if you smoke, right? But it's not something that is synthesized in the body. It's a substance that you consume from the outside, and then but still activates the receptor, nicotine, and experimentally was found to activate specifically this receptor, and hence was called the nicotinic acetylcholine receptor. It's a ligand-gated channel that admits both potassium and sodium, non-specific cation channel. This right here, the postsynaptic cell, either a muscle or a neuron. In this case, it's a muscle. Some experiments show that release of one acetylcholine vesicle from the presynaptic site will result in the opening of about 3,000 ion channels postsynaptically. To get a feeling of some numbers of this process. Okay, let's zoom into the structure and a little bit about the function of the nicotinic acetylcholine receptor. First of all, it's a pentamer. It's built of 5 unidentical units, which means that it's a heteropentamer. And upon binding of 2 acetylcholine molecules, the yellows right here, to these 2 sites, structural change occurs to the protein where each of these subunits rotates on the vertical axis, and this effectively you have a channel that's open right here, and allow sodium and potassium ions to pass. And different from the non-gated potassium channels with the specificity filter that were discussed last time, in this case, sodium and potassium ion pass with their shell of bound water molecules. So that's different. Also might be the reason, not might, definitely is the reason why it is not specific.
It's bidirectional, like,
It's bidirectional,
the sodium and the
effective,
potassium.
yeah, effectively I think the answer is yes, but when you open these channels, so due to their concentration gradients, they move really just to one side. So sodium moves in, potassium moves out.
Yeah, but
So
I
you
meant,
do
but
have
in,
two effects.
yeah.
Yeah, but still sodium, okay, so I guess it depends on the distance from the battery. And the distance from the sodium battery is higher, so I guess it emits more sodium, and hence will be the polarizing receptor, which it is. Right. This was very close to physiology, by the way. Oh, it's okay. Physiology at the single receptor level. Okay, the active substance in the cobra venom, interestingly, binds very stably, and when it binds, then it just stays there to the nicotinic acetylcholine receptor. It's thought that not necessarily 5, but anyway, it binds really stably to this receptor and blocks any transmission of ions through this channel. And it has other effects, but this is the main effect. And because the neuromuscular junction is cholinergic, so the first thing that happened after you chas v'chalila got bitten by a cobra snake is paralysis, right? So that's first. If it reaches the heart, then it's a real danger.
Shouldn't it reach the heart before it reaches the muscular junction?
No, because I'm not sure how the half-life of the substance, because of the pumping role of the heart. But still, if you're getting beaten peripherally,
Oh, yeah.
yes. Okay. Yeah, the paralysis starts from where you got bitten, I think, and then in the second stage it becomes systemic.
Because the parasympathetic system should be faster, right?
Yeah.
There's only one
It
side.
is.
So the heart will stop before your muscles will settle, right?
Yes, but what about this where the molecule reaches the synapses, the peripheral neuromuscular synapses first. So I don't know. Yeah, I'm not sure. Paralysis starts before. Is it? Yeah, I think yes. Okay. Nicotinic acetylcholine receptor, and that's it for the big chapter about different kinds of channels and pumps and so on. So just when you go over this chapter again at home, first of all, let's answer for next week. Please answer the question collection. I think I sent it to you by email today, if I have. And then when you go over the material, so make sure that for each gene family or product family, you know what is its main role physiologically. So for instance, as titled here, just make sure that you understand how the ATP pumps are the ones that maintain the intracellular ionic environment. And the non-gated ion channels, together with the ATP pumps, are responsible for resting membrane potential. This is the way to look at it. Okay. Now, so as you probably noticed, we're going straight until 12:30. Right. Next chapter is G protein coupled receptor, and it's parallel to Lodge's chapter, the 13th chapter. Right. And again, by now we've already learned enough to read the Kandel paper that I will send you by email. So you have 2 homeworks for next week. Answer the question page and read Kandel's paper. Do read the paper before we go over it together in class. Otherwise, it's really almost not effective at all. Just sorry?
You're gonna go over it.
Yeah, next week on Wednesday.
Kandel one?
But if you're not here, then we'll go over it together, okay? Anyone can comment later. So, yes?
Okay.
What? few general words about G protein coupled receptors. It's a huge family of proteins, really, really huge. Only 1,000 such genes code for G protein coupled receptors only in the human body, and there are many, many others across the whole eukaryotic animal kingdom. And they function in vision and in smell and in digestion and in many other processes. It's thought that all the ready FDA-approved drugs, well, not all, but about 60, 70, 80% of the available drugs today for humans function through GPCR. So this is a word of motivation. They are important, very much. You know which physiological system or which sense is the one that have the biggest array of GPCR?
Olfactory system.
Olfactory system, correct. So where you have receptor for each and every odorant in the molecule outside.
So, what was the answer?
Odorant, olfactory system is smell.
Mm-hmm.
So in the smell you have receptors, you have to have a receptor for each and every molecule, chemical molecule that's outside to be able to respond to it. And these receptors in the olfactory bulb are GPCR. I mean, G protein coupled receptors. Okay, let's start with putting the GPCRs in a general context. So when we look at the various signal transducing receptors that we have in the plasma membrane, we see an enormous variety of chemicals that can be involved here in several intracellular different mechanisms. The signaling molecules can be amino acids, lipid derivatives, small peptides, and the signaling molecules in each case act as a ligand which binds to a structurally complementary site in the receptor via a combination, as you see, and you probably got used to it by now, a combination of non-covalent interactions, right? Van der Waals, ionic, hydrophobic interactions. Anything that's not covalent. And the process of converting signals from outside the cell to something that will happen inside the cell to cellular responses is termed signal transduction. The whole process, not just part of it. The whole process is signal transduction. So in GPCR, we'll see that we have some receptor that recognizes a molecule. This is not the G protein yet, but it's a receptor that functions, that either activates or inhibits, usually activates, a G protein. So it's a G protein coupled receptor. And then the G protein, have some effect on some downstream effector. Okay, and then some of these processes can end up with synthesis of new genes, changing gene expression. Similarly for other receptor families, such as the cytokine receptors and receptor tyrosine kinases, these are also examples that function sometimes differently. Some of them have receptors even inside the cell, not on the plasma membrane, for steroids molecules that can pass the membrane. But generally speaking, we do see some similarities.
So this is what you call metabotropic receptors?
Yeah, these are metabotropic receptors, precisely. They do not open, they don't have any channel here. They will affect something will happen, and then another something, and part of these somethings will be an opening of an ion channel. For instance, in vision, and we will see. We'll start with some principles, the principles of intracellular signal transduction, principles of G protein structure and function, and then we will focus on these three classical systems for G protein, where G protein coupled receptors are the main factors. G protein coupled receptors that affect adenylate nucleotides, this is one pathway. The second one, G protein coupled receptors that regulate ion channels. We will learn about the vision pathway for road cells alone. G protein coupled receptors that activate phospholipase C. And then emphasize a few of the most downstream steps for these processes that sometimes end with gene transcription in the nucleus. Okay, so let's start with intracellular signal transduction principle discussion. So signaling molecules can be classified into three types based on the distance between the cell that secreted them and the cell that received them. Endocrine signaling is when the two molecules are, I mean, when the molecule was secreted and received between two cells that are very farther apart from one another. So they act on a target, the signaling molecules act on a target cell distance from where they were synthesized and secreted. So they have to be carried with something, right? Carried up with something. So usually they are carried by the blood, or in the central nervous systems by what we call CSF, cerebrospinal fluid, which is basically a filtrated blood. Very basic, because also other things. In paracrine signaling, the signaling molecule affect target cells only in close proximity. Neuronal synapses are an example of paracrine signaling, neurotransmitters, for instance, and also growth factors. These are cells that affect their neighboring cells, and not anything that's too distant. And autocrine signaling is when cell responds to a substance that it was the one that secreted it. So again, it's a bit weird, maybe, because why should a cell send the message? It's like sending a mail to yourself. Actually, all of us do that, right? Okay, but the system was designed to do so. Again, evolutionary-wise, or with evolutionary biologist glasses looking at it, we might say that endocrine and paracrine signaling were preceded in evolution. We actually do have some evidence for that. And then when the need for autocrinic signaling have emerged, it was based on what processes that were already available. Okay, an important emphasis. Some signaling molecules can act both short range and long range. Epinephrine, for instance, is a neurotransmitter paracrinic signaling molecule and a hormone, endocrinic signaling molecule. Same substance, right? Okay. External signals induce two major types of cellular responses. Either a change in the activity of some protein that's already there at the cytosol, changing its function, or changing the amount of a protein produced by a cell, increasing its degradation or its synthesis. Angioprotein coupled receptors can do either the one or the other or both at the same pathway. Subclasses of receptors can initiate signaling via more than one intracellular signal transduction pathway. Okay, so again, from the level of the receptors to the level of the intracellular processes, you have some convergence, because one receptor can induce more than one distinct intracellular process, right? But in parallel to this convergence between these two layers, we also have in parallel to these divergences, we also have convergence, because despite the huge number of different kinds of ligands and their specific receptors, a relatively small number of, possibly or potentially signal transduction molecules or mechanisms exist. What do I mean by that? So we said one receptor can activate several intracellular processes, but also a lot of different receptors or a lot of intracellular processes will end up increasing the level of cyclic AMP. So this is a divergence between these two layers, right? Convergence. So we have both. Okay, a word about specificity between the signaling molecule and the receptor. So each receptor binds only a single signaling molecule, and if you insist changing it, so just a few very closely related molecules, basically really just one single molecule, it was shown, and it will not be sensitive to bigger changes that will be done on the signaling molecule. It was shown on this signaling molecule, which is a protein, growth hormone, where researchers did point mutations, started changing mutations throughout the protein, and noticed when when you don't have any recognition with its receptor. So it's pretty specific region in the growth hormone molecule. But in contrast, a signaling molecule, just one signaling molecule, can bind to multiple types of receptors. And each time, it will activate different intracellular signaling pathways. An example, acetylcholine, if it is recognized by ligand-gated ion channel, in the muscle, it will be recognized by a nicotinic acetylcholine receptor, and it will cause muscle contraction via the intracellular process of ligand-gated ion channel opening. In the heart, it will cause slow of the heart rate via GPCRs, and in the digestive system, it will cause digestive enzymes to be secreted into the pancreas. I think also by GPCR. So these are different, right? Same molecule, different receptors, different intercellular processes. The intracellular processes are usually, in them you find involved what we call second messenger or secondary messengers. Why do they call second messengers? To who are they secondary? So the first messengers are the ligands themselves that came from outside the cell and were recognized by the membrane receptors, membranal receptors. So these are the first messengers, and their binding leads to short-lived increase or decrease in the low molecular weight molecules, second messenger. Examples of second messengers are cyclic AMP. Just leave the same structure and change the adenosine base here to a guanine base, and you cyclic GMP is the second messenger as well. Diacylglycerol, derivative of a phospholipid, and inositol, some kind of sugar, are also examples of second messengers. Usually low molecular weight molecules.
So what's the difference between regular AMP and cyclic AMP?
Regular when linear. So it's not linear. The phosphate here will not covalently link this carbon in the ring base.
Okay, not this.
Yeah, so it's cyclic. There's an enzyme that produces this cyclic form in the next couple of slides. Calcium is also a second messenger, the tiniest molecule, second messenger you'll find. And again, examples of the various inductions that a single secondary messenger can induce. So in muscle, a rise in cytosolic calcium triggers contraction. In endocrinic cells, it triggers exocytosis. And also in nerve cells, exocytosis of neurotransmitters, and so on. So I think basically, as is the case for neurons, where we are not always or not only interested in the input that is collected into a single neuron to understand its output or how would it respond, but also the important question is in what state do we catch these neurons. So a similar thing happens here. Which kind of neuron you have encountered and what is its state, and then the input will be, we could convert it into output or understand better the output. Okay, activation of all cell surface receptors. It's true for all surface receptors. Leads to changes in protein phosphorylation through activation of proteins. These are the proteins that add phosphate groups to other proteins, kinases. And these are the ones that take off phosphate groups from other proteins, phosphatases. About kinases, just a little bit more biochemical information. We have two kinds of kinases, those that add phosphate to hydroxyl group on tyrosine residues, these amino acids, and then the second type that adds phosphate to a hydroxyl group on either serine or preamine. Sounds like just these two are distinct structural and also functional. So these are really two distinct kinase families. Now each protein kinase phosphorylates specific residues in a set of target proteins, but each target protein can be substrate to multiple kinases. So it can be target protein X can be phosphorylated by PKA, PKB, and PKC. PKI is protein kinase. A lot of times you'll find that single transduction components are localized to areas on the membrane in the postsynaptic site. So this is how typically a postsynaptic site will look like. And this clusterization is achieved by adapter proteins that contain multiple protein binding domains. And the most well-established domain in this context is the PDZ domain. It's combined of 90 amino acids, this domain, and it binds to 3 residue sequences at the C terminus of target protein. This is the PDZ target protein. So it has some consensus sequence in the C terminus that is very, very, from what was known, at least for several years, it might be updated right now, but the target sequence, the consensus sequence on the target protein is very non-specific. Should be either, it looks like this, should be either serine or threonine, any amino acid, hydrophobic amino acid, or even if you try, you'll find a enrichment for hydrophobic amino acid, any amino acid, hydrophobic amino acid. Again, very non-specific. I think there later in the literature people talk about many other sequences that surround this sequence. But the PDZ domain, again, is found in many proteins at the postsynaptic site, mainly in the PSD95. This is the name of a protein. Look, the PSD is that's how it looks. It has multiple PDZ domains where other target site prosynaptic proteins can be anchored on. For instance, the NMDA receptor, another very important postsynaptic protein neuroligin, another kind of channel. And through additional adapters, the whole thing will be linked with the actin filament network down beneath the plasma membrane. Okay, so PDZ is the important domain to remember here that through which we achieve this clusterization, and the PSD95 protein is the one that has this domain. Okay, we discussed signal termination in neurotransmitter sinus cells. About GPCR, signal transmission is achieved via degradation of secondary messenger, right? Either taking the cyclic AMP and making it linear, or deactivation of signal transduction protein. But short range, if we talk about short range activation, there is no deactivation of the receptor. On the other hand, high signal concentration, external signal, or prolonged exposure to a signal cause receptor desensitization. So this is where we move really from the domain of short-term activation to long-term termination, or short-term termination to long-range termination. Again, when we talk about signal termination that's direct, we do not talk about receptor desensitization. If we cease receptor desensitization, that means we had a more than usual high signal concentration. Receptor desensitization can occur by receptor endocytosis, removing the receptor from the plasma membrane, receptor inability to bind the ligand, changing its structure so it will not be able to recognize its ligand anymore, or let it recognize the ligand but form a complex that does not induce a response, blocking the response through the receptor. Okay, so these were the principles about intracellular signal transduction. Now let's see the basic structure and function of G proteins, for those who are still surviving. Okay. G proteins are members of a bigger family that are called GTPases. These are proteins that hydrolyzes GTP, guanidine triphosphate. They are tethered to the membrane by covalently attached lipid molecules. And so we have trimeric G proteins, and we have monomeric G proteins. We'll see the difference between the two in a minute. But for the trimeric G proteins, these are proteins that have three subunits that form them. So, and these subunits are alpha, beta, and gamma. For these trimeric G proteins, during intracellular signaling, the beta and gamma subunits remain bound together and tethered to the membrane, and the G alpha is tethered by itself to the membrane and dissociates or shuttled laterally through the membrane. And this is really the part of the protein that is a GTPase, that has the hydrolysis capability. Right. We'll see better figures of trimeric G protein than this. So okay, let's see this. Let's mention several regulatory components of G proteins functioning. So the rate of GTP hydrolysis, again, the G alpha hydrolyzes GTP to GDP, and the rate is dictated gap, whose activity also controlled by extracellular signals. Again, get the feeling of how the state of the neuron is really, or a cell is really important to understand how the input becomes an output, or what happens with the input. So even the regulator is controlled by extracellular signals, so it makes things, predictions pretty hard to make. Activation of GDPs is mediated by guanine nucleotide exchange factor. After the G alpha hydrolyzes GTP to GDP, the GDP is taking out and a new GTP is taking in, and this is regulated by the the guanine nucleotide exchange factor, GAP. And there are two classes of GTPase switch proteins, as I mentioned in so these are the two, the trimeric G proteins which directly activate certain receptors, certain receptors, and the monomeric G proteins, which are all homologs of a protein called RAS, so we call them the RAS family. They are homologs to the alpha subunit, the single subunit, the alpha subunit in the trimeric G protein. And these two different G proteins are different in many ways, structurally and functionally. Okay, we're looking right now at the monomeric G protein, which look like the alpha subunit of the trimeric G protein. And in it, you find two regions called switch 1 and switch 2, two segments in the protein, which upon GTP binding, they undergo conformational change in these proteins, and then that's what allows the protein to bind and activate other downstream signaling proteins. And we call it the on state. So this is when the protein induces some downstream effect on a downstream effector when it holds GTP in its nucleotide binding site, and the switch 1 and 2 interact with this phosphate, the third phosphate. When it hydrolyzes the GTP to GDP, no interactions between the two switch domains is sustained anymore, and this is the off state. And in this state, either the RAS or the alpha subunit of the trimeric G protein does not activate any downstream effect. Okay, usually the effector protein, meaning the protein that is affected by the G alpha or the monomeric RAS, is activated by the G alpha. In some cases, it is actually inhibited by it. Also cases where the beta gamma complex is the one that either activates or inhibits the effector. In other words, all options are open. But again, the main thing that you find is that the G alpha is the one that activates or inhibits a downstream effector. Now, a trimeric G protein is not a specific protein. It's potentially composed of several combinations of alpha, beta, and gamma, and only the human genome encodes for 27 genes that code for G alpha, 5 G beta, and 13 G gamma options that you can choose from to build your own trimeric G protein. This is just a table that describes for each type of G protein in which system we can find it, and which is the second messenger through which it acts. So, okay, regarding the sequence of events during an activation of a G protein, so this is how it looks. This is the receptor, the G protein coupled receptor, the G protein trimeric form, this is the alpha subunit and the beta gamma. This is some downstream effect. Upon recognition of some signal molecule, in this case a hormone on the receptor, it's been activated again by some conformational change, activates the G protein. Up till here, the G protein has GDP in its nucleotide binding site, and now the GDP is taken out, replaced with the GTP. So now it's in an on state. And first of all, the alpha subunit dissociates from the beta gamma complex. Each each one has its own tethered lipid through which it's linked to the whole membrane, so it's not dissociated into the cytosol, it remains on the membrane, both the alpha and separately the beta gamma. And the GTP and the alpha subunit is the one that activates downstream effector, and it does so as long as it has the GDP in its nucleotide binding site. And when hydrolysis occurs, the G alpha becomes, hydrolysis occurs, you have GDP where you had GTP, and then the alpha subunit is not active anymore, stops activating downstream effectors, and rejoins with the beta gamma complex.
Isn't that
Brings us back to
process
the beta.
happens on the membrane?
Yes.
And then from the effector, I guess something moves on to the rest of the, like, from the membrane.
Exactly. So this effector will change the level of some intracellular molecule, and cytosolic molecule. This will
So
happen.
the first step is on the membrane.
Yeah, I don't know about the first. All these steps are on the membrane. Yeah, exactly. This is all on the membrane. We will skip the music. Okay, let's end with mentioning this important method. We have to trace the distance between G alpha and G beta gamma, and it will allow us to estimate pretty accurately what's the level of activation that happens on a G protein. It's called the Forster resonance energy transfer, or just FRET, and not as many mistakenly think fluorescence resonance energy. Although we have fluorescence in it, but it's named after this researcher. And in it we will see the kinetics of an alpha subunit dissociation. And as you understood by now, the alpha subunit dissociates while the G protein is activated, right? So this will actually tell us how much activation of a G protein we had in the system. Okay, so in order to implement this method, we have to link fluorescent dye to the alpha subunit, and separately a different dye to the beta gamma subunit. Then we excite the alpha subunit dye with a wavelength that will excite only this one, specifically this one. Upon close proximity between the two fluorescent dyes, you will have fluorescence energy transfer. This will happen quite effectively, and you will be able to read emission in this wavelength, in the yellow wavelength. And this is very not effective when these two molecules are farther apart from one another. So the signal will drop. If you plot the signal of the yellow, which is, we excite in the blue and then we read the yellow, and this is the fluorescence of the yellow light, normalized, right, from the maximum line. Upon ligand added right here, what happened when you added the ligand? You activated a receptor that activated the trimeric G protein, and then you read this drop in the yellow emission, which means that you have activation of a G protein. What do you think? In which of the two conditions, A or B, did you have more, which of the activation happened faster? A, right, the solid line, because it just happened, before, earlier in time, right? And which, I'm not sure you can say that because of normalization. Okay, so assuming you had the same number of G proteins in the system, in which condition, A or B, did you have, at the end state, more more robust G activation? B, right, because it's slower here, right? So you will read less of the yellow at the end state. Okay, so this was Forster resonance energy transfer method. And that's actually usually the case where it's a time scale of seconds. You can see how fast it happens. It's pretty much
How
depends
can
on
you
the
see that
system.
it's not at the same time?
Because the drop, this phase, happens earlier in the solid line than the dash line. Right? This happens faster. We had this weird drop before you even added the ligand, but I don't know. You just push it up.
So we're looking for the corner or something.
We're looking for this drop phase. Let's not define it
Okay.
mathematically for now. But
Sorry?
with no accurate mathematical definition, the drop here is a drop in the graph. slope, the downhill phase of this graph is when this complex started to be dissociated, right? So, yeah. Right. Okay. So we will end here, and then next time go into the specific example of receptors that act via a G protein. So last time that I'll remind you, read the Candel paper that I sent you and answer the question page for transfer of ions in other molecules. See you next Wednesday.